I'm Danny Yip. I'm a PhD student of Kevin Yip's lab, and it's my honor to have this opportunity to share our project, the systematic exploration of autonomous modules in the noisy miCoRNA network for testing the generality of the ceRNA hypothesis. We investigate the miCroRNA-target network and explore the modules systematically. The modules include the microRNAs and targets, and we use the modules to test the generality of the ceRNA hypothesis. And we have many recent studies related to the microRNAs, and we know that microRNAs can target some messenger RNAs. And then the targeted messenger RNA is replaced. In some cases, the messenger RNA is further degraded. And thus, it is expected to see certain correlations in the expression data and between the messenger RNA and microRNA. And we can find that many works make this as an assumption. And the real network, and indeed, it is very complicated, involving multiple messenger RNAs and multiple microRNAs and other regulatory elements. And microRNA targeting effects may not be directly observed, and we are not sure if the anti-correlation with assumptions can still be observed in the complex network. So we are going to verify this assumption by constructing modules using a large-scale data analysis and to see how general the ceRNA hypothesis is. The completing endogenous RNA hypothesis is proposed by ceRNA, and it is proposed that messenger RNA compete for the microRNAs, and thus there are certain buffering effects among the messenger RNAs. Let's take a look at this example. In the middle, it is the steady states. If we reduce the number of copies of RNA-Y and keep the same number of microRNAs, we can see that there are extent of inhibition of RNA is increased, and we expect the number of free copies of RNA-X will drop. On the other hand, overexpression of RNA-Y will lead to the increase in the number of free copies of RNA-X, and thus we expect to see certain correlations in the expression data, and we would like to see how general this hypothesis is. Let's take a look in this real network. It is constructed based on the union of 5 prediction algorithms. And we can see that one microRNA can target multiple messenger RNAs, and one messenger RNA can be targeted by multiple microRNAs. To obtain a simple modules from this complex network, and we use these modules to study the anti-correlation, the buffering effects, and it's not an easy nor a trivial task. There are several difficulties. First of all, there are many extensive linkages. If we have a nice group of microRNAs, messenger RNAs as shown here, it's very nice. However, in the real network, there are always extensive linkages. To solve this problem, some classical methods require lots of expression data to learn the models, or the computations, like estimating the parameters, is very expensive. Second, the network is very noisy, and we have very few validated pairs. For even a simple interactions between a microRNA and messenger RNA, different algorithms give different predictions. Third, in the whole network is very complicated. In addition to microRNA-target effects, there are some other regulatory mechanisms and indirect effects. Furthermore, if we use expression level of messenger RNA and microRNA to assess the microRNA targeting effects, it is not accurate enough. It's because where messenger RNA is inhibited by a microRNA, in some cases, the messenger RNA is not degraded. And the expression data, especially using sequencing technique, cannot account for this phenomenon. And potentially, there are some more difficulties related to the modulations. And here we show a list of previous work. This one. Okay. And on the microRNA target modulations of other research groups. And we found that most of them use expression data in the network formation. And in our work, we only use experiment interactions and predicted interaction to reconstruct the network. More precise, we use the validated interaction from TAR-based version 6 together with the top 5,000 predictions from 5 prediction algorithms to reconstruct the network. The expression data using to test the anti-correlations and buffering effects are extracted from the ENCODE project. As our goal is to verify the correlation relations, we keep the correlation expression data. We keep the expression data away from the network formation. And here's come to our solution. Finding autonomous modules, again without using the expression data. The modules contain several microRNA and messenger RNA, such that the modules are largely separate from the remaining network, and therefore anti-correlations buffering effects can be easily observed. And the networks can be modeled as a bipartite graph. Two sides refer to messenger RNA and microRNA respectively, and we only allow edges containing 2 different sides. Or the network can be modeled as a binary associated matrix. Each column means microRNAs, while each row refers to a messenger RNA. If the number is 1, it indicates there is an interaction. Here, this is an ideal case we are looking for. The groups of microRNAs target everyone in the groups of messenger RNA, and the groups of targets are consistent among both messenger RNA and microRNAs. If we show this is a good result, it's very clear signature showing the existence of miCoRNA targeting effects. And we call this kind of modules as miCoRNA-target bicluster, or this is a restrictive type, the type R MTB. However, we have mentioned the real network is very noisy. The restrictive type is very sensitive to the errors, causing very few numbers of cases. So we introduced some other types of MTBs. For example, we allow some messenger RNAs to be targeted by some extra microRNAs. And furthermore, we allow the microRNAs to target some extra messenger RNAs. And these 2 features enable some false prediction outside the MTB. On the other hand, we allow some microRNAs not targeting all messenger RNAs. These features promote misinteractions within the MTB and allow more imperfect cases. And similarly, we have more definitions, and altogether we have defined 8 different definitions. And it's not obvious that which definition is better. And after evaluating each type, we can perform some other empirical comparisons. And we evaluate the MTB based on the microRNA target network. Next, we have several computational and biological questions to study. Computationally, how can we find each type of MTB? How many MTBs of each type have? Biologically, can we verify the anti-correlation relations between the expression nature in messenger RNA and microRNA? Do the messenger RNA buffer each other? Do the group of messenger RNA share similar functions? And can we apply our MTB definition to some other problems? For the computational issues, we propose algorithms for finding MTBs. And we also analyze the algorithms of each type of MTB in terms of the number of solutions, the time complexity, and the MTBs returned by our algorithms. Let's briefly talk about one case, the type RGen MTB. Within the MTB, all microRNA and messenger RNA target each other. And outside the MTB, both messenger RNA and microRNAs can target some extra RNAs. And we can see that the groups of messenger RNA and the groups of microRNAs are fully connected. And identify this type. The type RGen MTBs is the same as finding biclists in the bipartite graph, and this is well-known anti-complete problems. Up to now, no polynomial time algorithm can solve this problem. The problem itself is proved to be computationally difficult, and we propose algorithms for finding this type RGen MTB. In practice, the graph or the matrix is sparse, and our algorithm is able to find all type RGen MTBs in a reasonable time. And next, we move to the biological questions. How can we verify the microRNAs and messenger RNAs in our MTB that are more anti-correlated than a certain background? First, we identify the MTBs based on the network, and then we will construct the background. And then we use the expression data to evaluate the score of each MTB. as well as the backgrounds. And we have the distribution of score of MTBs and distribution of scores of background. And having those distributions, we can calculate some statistical tests and test how significantly different between these two distributions. And we have a simple example here. Negative numbers in the table refer to the anti-correlation coefficient between the corresponding microRNA and messenger RNAs. If we say that the correlation value is more negative than negative 0.2, it is a negative correlations, and here we have 7 numbers out of 9 are negative correlations. And the scores of this MTB is defined as 7 divided by 9. And we have a list of MTB, we will have a list of distribution of scores. And we also have a distribution of scores of the background. And then we plot the distributions of the scores of MTBs and the backgrounds here. And we call that the scores is a fractions, so 1 is the best score. And we can see that our MTB, the blue bars here, outperform the background. And then we have the 2 distributions, and we can perform the statistical test on the anti-correlations. In this graph, x-axis is the thresholds we have used in the anti-correlations. In previous example, we have used negative 0.2. Y-axis is a p-value calculated in the statistical test. And we can see that our MTBs did a great job if we choose the thresholds ranging from -0.1 to -0.5. And different lines here refer to different types of MTBs. And we can see that the loose type, allowing more imperfect cases, did better job than the ideal cases or the restrictive types. And it means that we really require some error tolerance in defining the groups of RNAs. And to ensure the robustness of testing, we have tested different data sets and different backgrounds. Previously, it's a random background, and this is a background target pairs not formed by the same MTBs. And the result is also good. And next questions, we try to verify the buffering effect among the messenger RNAs. One proposal is to test the positive correlations among the messenger RNAs. However, positive correlations among the messenger RNAs can be simply a cause of co-regulations of other effects. This not necessarily come from microRNA targeting. So we come up with other proposals using the conditional correlations. R is the microRNA, T1, T2 are the messenger RNA, and R target on both T1 and T2. And if we know T2, having more information, the correlations between R and T1 would be more negative than without the knowledge of T2. So we try to test the distance between these conditional correlations with the simple distributions, simple correlations. And we found that both proposals give consistent good results, and we show that messenger RNA in the same MTB buffer each other. As we have selected many groups of messenger RNA in the MTB and the background, and we can perform gene set enrichment analysis using human gene ontology annotations. We also find that the groups of messenger RNA selected by the MTB have more functionally related according to gene ontology. A short summary, our MTBs allows us to observe clear anti-correlation between our groups of messenger RNA and targets. Our data analysis also supports the generality of ceRNA hypothesis, and it means that the messenger RNA within the MTBs buffers each other. And the groups of messenger RNA having shared common targeting microRNAs are more functionally related according to the gene ontology annotations. And now we propose our MTB definition to some other aspects. For example, if groups of messenger RNAs are related to certain pathway, the groups of microRNAs may also involve in the same pathway. And it's also interesting to ask if we can improve the prediction algorithm using our MTB definitions. The idea is like this. Interactions within the MTBs, we believe, is more confident than the interaction outside the MTBs. And these features can help improving predictions. And in addition to understanding the modules, it's also interesting to study the hierarchy of the modules, the relation between each pair of MTBs. And lastly, the MTB definition is is general. The definition only based on the interaction network, and thus we can apply our definitions to some other regulatory networks. Last but not least, many thanks to our collaborator in the Chinese University of Hong Kong, the Hong Kong Bioinformatics Center, and we would like to give a special thanks to Dr. Sarah Yabali for pre-processing the RNA-seq data. Thank you very much. Any questions?
Hi. Do you control for gene length bias in your tests?
Sorry.
Gene length bias. So I mean that longer genes are more likely to share microRNA binding sites just by chance, and they're also more likely to be co-expressed with each other. So you'll find that co-expressed genes share the same microRNA binding sites, but it can always be only due to gene length bias.
We did not include the length of the genes in the analysis, and we only used the interactions from the experiments and the prediction algorithms. And it is also another features of including the length of genes in the analysis. But in however, our methods is enough for determine the anti-correlation and buffering effects among the messenger RNA and the microRNAs. So if include more information, it's expected to see a better results, but it's okay now at this point.
So I'm a little bit confused about the final goal of this analysis. So, there's a number of publications that come out, including our publication in Cell about a couple of years ago, that show that you can get very large clusters of ceRNAs, that they are very highly correlated, and in fact that their correlation increases with the cluster size. There's been a lot of biochemical validation showing that these interactions are Drosha dependent, et cetera. So in terms of showing that the ceRNA hypothesis is reasonable, I'm not sure how the MTB is actually increasing that. And then what I don't understand is, so the MTB selects essentially a core module of conserved microRNA-targeted conserved set of ceRNA, but why is this more important than simply the fact that two ceRNA share a large program of microRNA that couples them? So in your case, in your analysis, for instance, this will be missed. You will only identify these subprograms where everything is actually shared in a very similar way, right? So by co-clustering. Well, I would say the important thing is to know whether a particular ceRNA is inhibited by another or is modulated by another or not.
Can you repeat your question? It's too long. It's too long.
I'm just saying, right now what you're doing, you're creating a matrix where all the miRNA and all the mRNA are sort of looked at in potential interaction, and then you're selecting by clustering a core, what you call an MTB, right? I'm asking, why is that more important than simply knowing whether a particular mRNA and another mRNA interact because they share a large microRNA it didn't quite come across as an explanation, and you may miss, by using the MTBs, you may miss a huge number of interactions that are very relevant from a biochemical perspective.
We are interested if the multiple microRNAs, multiple messenger RNAs that target each and others, and because in the microRNA target levels, and it's so complicated, and we try to have this kind of
certainly use enriching for some features, like co-expression, buffering, function. So I guess the question is, do you better enrich for those features?
So extending on the same thing, I have a quick question. So have you actually tried to look into the binding sites of these microRNAs to see if they overlap? Because you could think of at least two hypotheses. MicroRNAs are competing for the binding sites on the target transcripts, or they're collaborating and together they actually repress. Have you tried to take this into account?
In most previous work, we looked at most microRNAs when it's target on the messenger RNAs will inhibit the messenger RNA and stop the protein translation. And it's very few related work say that it will upregulate or some other effect.
No, I didn't say about the upregulation. I'm talking about the binding pockets on the target transcripts. Anyway, we can talk about it later. A related thing is